The age gap in cancer research. Why studying young models may be hindering effective treatment, a fundamental mismatch in cancer research is potentially stalling medical progress. While the majority of cancer patients are older adults, the vast majority of laboratory research is conducted on young subjects. This discrepancy creates a significant gap between successful laboratory results and real-world clinical outcomes. The youth bias in laboratory science. Current cancer research relies heavily on young mice, which are biologically comparable to humans in their early 20s. According to recent data, fewer than 10% of cancer experiments include aged animals. This reliance on young and fit models is driven by practical constraints. Cost. Younger mice are significantly cheaper to maintain. Time. To study aging, mice must be raised for 18 to 24 months, a lengthy period for many research cycles. Simplicity. Young mice possess healthy, intact immune systems that are easier to study in a controlled environment. However, this bias creates a translation gap. Therapies that appear highly effective in young, healthy models often fail in human clinical trials because they do not account for the complex biological realities of older patients, who frequently face different immune responses and higher toxicity risks. New findings. The nonlinear relationship between age and cancer research, presented at the American Association for Cancer Research annual meeting by the Fox Chase Cancer Center, suggests that cancer progression does not follow a straight line as we age. In a study focusing on melanoma, researchers discovered a surprising pattern regarding how cancer spreads. Young mice showed the lowest rates of cancer spread. Middle-aged mice experienced the highest rates of metastasis to vital organs like the lungs and liver. Very old mice, surprisingly, showed a decrease in cancer spread compared to the middle-aged group. The role of \gamma\delta $T cells, the key to this phenomenon, appears to lie in a specific group of immune cells known as gamma delta. \gamma\delta T cells. These cells act as an early defense mechanism against cancer. The study revealed a distinct correlation between these cells and age. High defense. Both young and very old mice maintained higher levels of \gamma\delta T cells, which helped keep tumors dormant or localized. The middle-age vulnerability. Middle-aged mice had significantly fewer of these protective cells. Furthermore, the study found that in this age group, melanoma cells actively released molecules designed to suppress or exhaust the immune system, allowing the cancer to spread aggressively. Bridging the gap. New tools for aging research to combat the lack of data on older subjects. Researchers at Fox Chase, including Mitchell Fain, PhD, and Yash Chabra, PhD, have established a dedicated aged mouse facility. By creating established colonies of older mice, they aim to lower the cost and time barriers that previously discouraged scientists from studying aging. This facility allows researchers to move beyond one-size-fits-all models and begin asking critical questions. Why does cancer risk seem to drop in patients over 85? And how can we protect the immune systems of middle-aged patients to prevent aggressive metastasis? Understanding how therapies affect older patients would give us more and better treatment options, says Mitchell Fain, PhD. Conclusion. By shifting research focus from young models to aged subjects, scientists can better understand why cancer behaves differently across the lifespan. Addressing the age gap in research is essential for developing personalized, effective treatments that work for the actual demographic most affected by the disease.